Hi, Mike Gibson, Dean Kereiakes, coming to you virtually from TCT 2020. Dean, gosh, 25 years ago, we used to have a balloon, the Ace balloon and the Pro balloon, getting out in those diagonals with a balloon that had a little wire on the end of it. A lot of the fellows these days aren't old enough to remember those balloons. It was always great to get to those tough spots. And now you have a balloon with a stent on it with the same kind of design. Talk to us a bit about the Optimized trial.
Well, thanks a lot for having me, Mike. Optimized is a pivotal trial, IDE trial for FDA approval of the Svelte IDS, which is an integrated delivery system. It literally is a stent on a wire using Asahi wire technology. And it's RX version, which is the monorail version of the device. This is a very interesting device. The 2.5 millimeter IDS, which is the integrated delivery system, is a 31,000 profile, and the Trio stent is a 32,000. So these are smaller than the wires we use for vascular access. They're mounted on a low compliant balloon, or virtually non-compliant balloon that uses folding technology, proprietary technology to reduce the profile. And the actual strut thickness, it's a cobalt chrome platform, it's 81 microns. But it has a unique polymer. It's an amino acid composite that undergoes enzymatic erosion, not hydrolysis like the PLA derivatives, over about 12 months. And it's very anti-inflammatory. There's a marked increase in IL-10, the anti-inflammatory cytokines, and a marked decrease relative to other polymers in IL-6 and the inflammatory cytokines. So I think that's one of the unique aspects of the device too, is this novel drug coating, which is the polymer that elutes sirolimus. Okay. Mm-hmm. This is a pivotal trial. It's a classic non-inferiority powered based on Evolve-2. In fact, that's the interesting twist. I was the PI for Evolve-2 also, which is a pivotal trial for synergy approval. This basically HCRI, Laura Mori at that time, took Evolve-2, said we had a 6.5% TLF rate at 1 year in the control population, which is Promus element. And we're going to power off of that with a non-inferiority margin of 3.58% at a significance level of 0.025. So if you use that as a benchmark, and other DES trials have since that time, the 8% TLF has come down to 6.5%. But one thing that you should know is that 99% of that control population was assessed with CKMB, not troponin. Okay. And in retrospect, that is a very important fact. So we powered this and had to get at least 1,630, and there are 1,639 patients that were randomized between Promus or Xience on one hand, that is the control, which are, PVDF, polymer, everolimus-eluting, permanent polymer stents, versus Svelte on the left, whether it was IDS or RX. And at 1 year, the TLF rates were 10.3% Svelte, 9.5 Xience Promus. So the absolute difference, the upper confidence bound of that absolute difference is 0.8%, actually slightly crosses the non-inferiority margin. P-value of non-inferiority is 0.034, which doesn't quite meet the
0.025.
Level of significance. But here's the rub, as we say, Mike, and it's very important. I think this trial will inform all future DES trials, very frankly, for the following reason. 25% of our patients were assessed solely with troponin. And if you look across both devices, whether it's the test device or control, troponin I, we're here at my hospital. You know what the upper limit of normal is?
One molecule or something.
No, less than 0.05.
Yeah.
Okay. Less than 0.05. It's 0.03. So an MI is 0.09. This is it any surprise that almost 33%, between 30 and 33%, didn't matter which device, has a TVMI
Right.
population. Troponin T, 10%, regardless of device, have a TVMI.
Mm-hmm.
CKMB in the 75%, less than 2.5% have
Yes.
TVMI.
Yeah.
So when you do a reanalysis using sky definition, which accommodates troponin, the threefold was never designed to accommodate troponin, frankly. And we couldn't foresee the evolution in the American hospital system in particular, where everybody now doing troponin, and many places don't even do CKMB.
Yes.
So, it really puts a wrench in the TLF as the endpoint, and it ends making us effectively underpowered. So if we powered the trial post hoc now in retrospect, which I understand we be critical of, we need almost 5,000 patients in this trial. So I think the answer is we are non-inferior if you do it with a sky definition, highly significantly non-inferior. If you do it with a relative non-inferiority margin, which anyone would use now,
Right.
in retrospect, and the trial definition for MI, you are highly significantly non-inferior. Stent thrombosis rate is less, 0.38 compared to 0.51, not significant, but numerically less than the Science Promise. The TLR rate is 1.5 was felt in 1 year. So, this is numerically less than Science Promise, maybe the lowest ever reported, frankly, in an IDE trial for, 800 plus patients. So I think the device itself performed extremely well on all parameters. I think the primary endpoint, as defined prospectively, is, I won't say erroneous, but it's obviously got limitations now that we know and could not have predicted that there'd be 25% of the patients were solely evaluated with troponin. They were 80% of all the target vessel MIs in the entire trial were in that 25% that were evaluated solely with troponin. So, if you were going to go forward with an ID trial, let's say you're going to design one going forward today, you would either create a definition that accommodates troponin, or you would go with a relative non-inferiority margin as an appropriate strategy. So I think going forward, this is going to raise the eyebrows of everybody designing IDE trials.
Absolutely. And, if your objective performance criteria is based upon trials that did not have that high use of troponin,
100%.
you're sunk before you even get started, right? You just can't outrun it.
Absolutely. Somebody was going to step on the landmine. That's what.
Yeah. Yeah. Well, hopefully the regulators will look at this closely, and hopefully this will inform, as you say, Dean, regulatory science moving forward, and hopefully they'll look at the totality of the data rather than just one endpoint.
Yeah, no, I believe so, because in retrospect now the issue is obvious at this point, and it's not an issue of the device; it's an issue of the biomarker. That's what it is.
Right. But Dean, thank you for sharing this. It's a complicated story, and it's something that all of us as trialists and practicing clinicians need to understand how complicated it has gotten. Hopefully, we can all work together to create a clear path forward in evaluating breakthrough technologies. Thanks for taking time out, Dean, and thanks to all of you for joining us here virtually from 2020. Thanks, Dean.